and he will present on a novel approach to potentially treat influenza, a selective induction of apoptosis in infected cells by hijacking the virus machinery.
Okay, thank you very much, Annemieke, and thank you, ASGCT, for making this meeting possible. Here are my disclosures. And influenza, as you know, is still a global health issue. It infects about 1 billion people every year, and it kills almost half a million people annually. It is caused by a negative-stranded RNA virus, influenza virus, and it facilitates a protein synthesis and replication through its own polymerase enzyme, which is an RNA-dependent RNA polymerase. And the way the DRP works is it uses the negative sense viral RNA as a template, and it produces a positive sense mRNAs from the negative sense viral RNA. And the way the polymerase enzyme recognizes the viral RNA is through the non-coding sequences on both ends. These non-coding sequences are important when it comes to our mechanism of action. We call this our novel mechanism of action hijacking the virus machinery. It is essentially the delivery of an engineered molecule, an oligonucleotide that is designed to be recognized by the viral polymerase and processed to express a kill signal protein or any protein of interest for that matter. And in an uninfected cell, because the viral polymerase is required for our oligonucleotide to be processed in an uninfected cell, our oligo is basically non-coding or non-functional. In the context of influenza, how hijacking the influenza polymerase works is basically, you can see on the bottom left, our hijack RNA carries the non-coding sequences of influenza RNA. So we basically trick the influenza polymerase into believing that our hijack RNA is a part of the virus itself, and it has to be used to produce mRNAs from our RNA. The open reading frame of our hijack RNA is basically the negative strand of caspase 9 coding region. So what it turns into after it's processed by influenza polymerase, our hijack RNA produces mRNAs that code for caspase 9 protein, and essentially the caspase 9 overexpression ultimately kills the cell. In the interest of making this easier, we produced AAV particles that express our hijack RNA, and we ran multiple experiments with MDCK cells infected with multiple different strains of influenza. We also wanted to see if our hijack RNA works on different strains of influenza virus. We had a GFP expressing AAV as a control. We treated infected cells with either virus and monitored caspase 9 expression, cell viability, and the influenza infection percentage in the cultures daily. So when we look at the caspase 9 expression in uninfected cells, as you can see a very low baseline of caspase expression in uninfected cells. And you can also see that our hijack RNA had no effect in caspase expression in uninfected cells, which means, as we expected, it was inert in the absence of influenza polymerase. When you look at the infected cells on the graph B, the blue bar shows untreated infected cells. You can notice that there is a natural increase in caspase 9 concentration in the cell. This is due to the influenza infection. And when we add our hijack RNA AAV into the system, you see that there's a threefold increase in caspase 9 amount expression in the cells, which made us believe that our hijack RNA, in fact, was processed by the influenza polymerase and produced caspase 9. And when we look at the cell survival curve over five days, this is the uninfected cell culture. As you can see, the hijack RNA AAV had no effect on the growth of uninfected cells. In the infected cell cultures, when we administer hijack RNA AAV, about 24 hours after influenza infection. Within 2 days after the AAV administration, we see a substantial cell death in infected cells. So the infected cell curve, untreated cells are the red graph, and the green is treated infected cells. And by hour 72, there's a dip in the cell survival. There is a significant killing, and we see the same effect across actually 3 strains of influenza. And after 72 hours, the cells picked up growth. So we wondered what happened after 48 hours. Did our hijack RNA stop working, or did it eradicate the infected cells? And all the surviving cells in the culture are uninfected cells, and that's why they picked up growth. If you look at the blue lines, that's the uninfected cell growth. So the fact that the last 2 days, the green and the blue lines seem quite parallel, this made us believe that we actually eradicated the infection, and that's why the cell proliferation picked up parallel to uninfected cell cultures. To make sure that's what is exactly happening, we monitored the infected cell ratio in the cultures with influenza nucleoprotein staining and flow cytometry analysis, and this is what we saw on the graphs on the right side. Across 3 strains within about 48 hours after hijack RNA AAV administration, over 90% of the infected cells were dead, and somewhere between 72 and 96 hours, the rest of the cells were gone. So within 3 days, the hijack RNA AAV completely eradicated influenza infection in our cell cultures across 3 different strains. So the next question was, how did this happen? Was it strictly by the hijack RNA treatment, or were there other factors that we were not aware of? We tested a hypothetical mechanism of action by adding caspase-9 inhibitors into the system. So this is what we expect to see. The caspase-9 inhibitors would neutralize the caspase-9 functions, and we would expect a growth pattern that's parallel to infected untreated cells, and this is what we saw. So the caspase-9 inhibitor group is the purple line, and the infected untreated cells are the red line, and you see the growth pattern or the death pattern is quite similar. So caspase-9 inhibitor, in fact, abrogated the effects of our hijack RNA AAV. And you can also notice that there's a slight increase in the cell survival in the caspase-9 inhibitor group. We think this is because, if you remember, there was a natural increase in caspase-9 expression in infected cells due to influenza infection. So we think the caspase-9 inhibitor that we added into the system was not only enough to neutralize the caspase-9 that is produced by our hijack RNA, it also neutralized some of the caspase-9 that was endogenously produced as a reaction to the infection itself, and that's why the cells survived a little better compared to untreated group. So to sum it all up, influenza, a global health issue still, despite vaccine and treatment options. We came up with a novel mechanism of action to hijack the virus machinery and express caspase-9, which successfully eradicated infections within 3 days across 3 different strains of influenza, and it had no effect on uninfected cells. We confirmed our mechanism of action with the caspase-9 inhibitor added to the system, and we're envisioning this to be an RNA nanoparticle drug, maybe as an aerosolized or nebulized delivery for respiratory system, and we're hoping this could be potentially an effective pan-influenza therapy because of the fact that our hijack RNA is processed and able to hijack all different.
Work at a higher level, if you used adenovirus for delivery rather than AAV? And then the second question related to that, the adenovirus would be transient, so it would last for up to 2 weeks, while AAV might persist longer. And if you're talking, we can't hear you.
Epithelial cells in the respiratory system, so it's easy to.
Oh, sorry. So, well, maybe it's just me, but I only just heard you again. So you'll have to re-answer the question.
Okay.
Sorry about that.
Okay. We envision this as an RNA drug because influenza, as you know, infects the respiratory cells, epithelial cells in the respiratory system. So local delivery is quite easy. And, you know, AAV has immunologic implications. Adenovirus has immunologic implications. So that's why, and also, you know, as a cost effective treatment, both viral vectors are quite expensive, so you can't imagine using it for influenza. But RNA drugs are a little more practical. But for other viruses, for hepatitis B, for example, it is an AAV redesigned and tested. And to answer your question, we are also working on SARS-CoV-2 with the same principle.
That would be an obvious question, of course, but it's good to hear that you are. Then I think other questions are coming in. So do you see, what about inflammation? Do you observe a drop in cytokine profiles with your treatment? So
this was only in vitro. You know, influenza virus is a cytopathic virus, so infected cells are destined to die. The way this works, we believe, of course, we're killing the infected cells that are supposed to die anyway, but we're also preventing the spread of infection by killing the infected cells along with the virus in it. So I don't expect any inflammatory response higher, you know, more intense than the influenza disease causes itself. But it is, of course, possible. It depends on how extensive the infection is spread in the system.
So that would be something that you can see when you go to the in vivo models, of course.
Yes.
Yeah. Okay, so thank you very much for your talk, and apologies about the technical issues that we had. So my laptop also has some technical issues. I can't seem to turn on my video again, but you know by now what I look like, so that doesn't really matter.
Neither can I. That's what I tried to work. It's fine. Thank you.
So, sorry you have to miss our faces, but I'm going to introduce the last speaker of this session already, who is John Lewis from the